Welcome to the last topic of the midterms. Genetic disorders. In the study of genetic disorders, it is very important that you remember the central dogma of molecular biology, the concepts of mitosis and meiosis. There are three categories of genetic disorders, and in this module, the focus will be mainly on numbers 1 and 2. First, mutations that results in single gene disorders. But what is mutation? It is defined as permanent change in the DNA, and a mutated gene can be inherited or can occur in adult cells when exposed to something mutagenic. In this module, the focus will be on point mutations within coding sequences and deletions and insertions. In point mutations, there is missense and nonsense mutation. In missense mutation, it is either conservative where there is no obvious signs and symptoms of the mutation, and non-conservative, where sickle cell anemia is an example. In sickle cell anemia, the amino acid glutamic acid has been replaced by valine in the 6th position of the beta-globin chain. The next slide will show illustrations of the mechanism behind the genetic disorder sickle cell anemia. In this slide, is an example of nonsense point mutation. This slide shows an example of a mutation called frameshift mutation, which occurs when there are insertions or deletions of multiple nucleotides. As seen here in the genetic disorder Tay-Sachs disease, which involves the enzyme hexosaminidase, that is needed to metabolize neurotoxic gangliosides. Briefly, we will discuss Mendelian disorders. Remember the monk who is also a biologist, Gregor Mendel, and his experiments with peas. His experiment with the peas helped in the understanding of the patterns of inheritance. Which will be the dominant trait? Which will be recessive? Is it autosomal? Or X-linked? Hence the patterns of inheritance of diseases. Autosomal dominant, autosomal recessive, and X-linked. The following slides will present disorders that are examples of each. Let's now briefly discuss chromosomal disorders. Karyotyping is the study of chromosomes. The chromosomes are counted in total, then followed by the sex of the individual. If there is an extra chromosome, as in trisomies, it will be written as shown in this slide. Chromosomal disorders may be numerical or structural. Numerical disorders may happen when there is a trisomy, as in trisomy 21 or Down syndrome, and in structural, there could be deletions, translocations, as seen in CML or chronic myelogenous leukemia, between chromosomes 9 and 22, or inversions. We end this brief discussion with some examples of tests that can be used in the diagnosis of genetic disorders.